Welcome to OmicsBox, Bioinformatics Made Easy.
All right, thank you for the introduction, Stefan. And with that, we're ready to begin today's webinar on the long-read transcriptomic analysis of hexaploid sweet potato in OmicsBox. So the dataset that we will be analyzing today is actually novel data that comes to us from one of our collaborators, Dr. Mingao, at the Prairie View A&M University in Texas. Thank you very much for agreeing to let us use your data in this little showcase. So the dataset is, of course, hexaploid sweet potato, sequenced with Oxford Nanopore direct RNA, and we have in total 19 samples, which are split across one parental line and 7 different offspring lines. And thanks to a recent publication in Nature Plants, we also have available a phased hexaploid reference genome and annotation, which we will be making use of in our analysis. So the study objectives for this webinar are to learn how to deal with biological replicates in long-read RNA sequencing data, to use such data to discover novel transcripts, as well as curating your custom transcriptome in order to improve data quality. And finally, we will quantify our data on this curated transcriptome and use the quantification to conduct differential expression analysis. So the workflow for today's webinar will start with transcriptome reconstruction through FLAIR. Then, as we will be conducting transcriptome reconstruction for each line of our sweet potato individually, we will combine the resulting transcriptomes using TAMA merge. Finally, this transcriptome we will conduct quality control and curation with scanty-free before quantifying the curated transcriptome using, again, FLAIR. And once we have obtained our quantification, we can conduct differential expression analyses. Now, all of this we will do through OmicsBox, which offers a very convenient and easy-to-use graphical interface, meaning that users have no need for any coding expertise. And thanks to cloud-based execution of all of these different steps, it is also not necessary to have any dedicated hardware. Now, to go into a bit more detail on all of the different tools that we will be using today, let's start with the first step, using transcriptome reconstruction with FLAIR. So FLAIR will help us to identify as well as quantify both known reference as well as novel isoforms from our data. So this step of transcriptome reconstruction will go from the reads all the way to transcript models, to a transcriptome. And while there are many different tools available, even in OmicsBox, to do this step, today we will choose FLAIR as, according to recent benchmarks such as the Longer Gasp challenges, it excels at the discovery of novel isoforms. So here our goal is to obtain a custom transcriptome to discover novelty for each of the lines individually in order to discover novel transcript. So as we will be conducting this transcriptome reconstruction step on each line, we will then need to combine these transcriptomes into a more general sweet potato transcriptome, which we will do with TAMA merge, which is a tool which facilitates this, what we call a call and join strategy for sample combination. This actually relates to my own research, so we have a manuscript on this in preparation, where we look into different strategies on how to combine biological replicates for long-read RNA sequencing data, specifically relating to transcriptome reconstruction, as the identification of novel isoforms is a crucial step in any such analysis and can have effects on any downstream analysis. So we're trying to go into more detail to evaluate different such strategies. So for the call and join strategy that we have chosen for today, we will run FLAIR either on each sample or, in our case today, on each line individually, and then combine the resulting transcriptomes, which the advantages of this strategy is that it is more flexible and efficient. So we can stop our process anywhere halfway through. If we get new data from new lines in the future, we don't have to redo any analysis on the existing data. Now, some disadvantages are that it may miss particularly lowly expressed novel isoforms. So if our study objective was to really dive deeply into any potential novelty, no matter how lowly expressed, then this might not be the best approach. But since here our ultimate goal is to conduct differential expression analysis, any particularly lowly expressed novelty will probably not even be relevant for differential expression due to the low expression. So we're willing to sort of deal with this disadvantage for our goal today. So again, we want to combine our transcriptomes into a general transcriptome, which will, of course, contain a lot of novelty, some of which may actually be the result of artifacts, either on the biological or on the technical side. So in order to understand what our transcriptome looks like, as well as to curate it to improve our data quality, we'll be using SCANT-free, which allows us to compare our custom annotation to a reference annotation, as well as to orthogonal data. And further, it can filter probable artifacts to obtain a high confidence transcriptome. SCANT-free can do this either via a machine learning filter, which is particularly effective when you have access to different orthogonal data, or via customizable rules, which is the path that we'll be choosing today. As with our non-model organism, we might not have many different types of orthogonal data available. So our goals with SCANT-free today are to understand the novelty in our custom transcriptome, as well as to curate our transcriptome to improve its quality. Then, after quantifying on our curated transcriptome, we will conduct differential expression analysis with EdgeR. So we will compare the expression levels between different groups of samples, depending on different traits or different lines, in order to identify statistically significant differences and potentially identify genes or transcripts that may contribute to differences between our different groups or different lines. So with this, we're ready to start with our analysis in OmicsBox. I've already loaded up all of the data we need. So again, our first step now is to run FLAIR in order to conduct transcriptome reconstruction, which we will find in the Transcriptomics module. Under long-read analysis and transcript identification, we will find identification and quantification with FLAIR. So as we are conducting this step on each line individually, I will load here only the long-read files from, in this case, the parental line, and then repeat this step for each of these 7 offspring lines. So we load only the data from one single line. We load our reference genome, which we have available, and then we configure our analysis. So, of course, we want to conduct transcriptome reconstruction. We want to identify novel transcripts, as well as quantifying our reads. So this will be a preliminary quantification, but we will also already want to have a bit of a measure of how many supporting reads each transcript has. So we also have a reference annotation available that we will be using. And further, in order to be able to report novel splice junction, FLAIR actually requires the use of short read data, which is a way of using orthogonal information in order to have more confidence in the novel splice junctions that we are observing in our long reads. Since we don't have now, well, we would recommend to use short read data here, but since we don't have them available in our specific experiment now, instead, I have extracted some high confidence splice junctions based on some custom filtering from our long read data, and we're using that as a stopgap here. So next, we can provide our own alignment files or let FLAIR do the alignment for us. Since the alignment here has to, we had to take a bit more care as we're working with a hexaploid organism with a phased genome assembly. It was necessary to take a bit of extra care to, for instance, keep in mind that we will observe a notable level of multi-mapping of reads that map equally well on different parts of the genome, for instance, different haplotypes. So we are providing our own alignment that we have previously created with Minimap2. Now we can configure our analysis in more detail with various parameters that are explained in the help tips or in our user manual. But for the most part, we are today fine with using the defaults. And of course, if later we review our results and we're unhappy with any of the results, we can revisit this step, conduct another analysis, and adjust these parameters. Finally, we select where we want to receive our files, and we're ready to launch our analysis. Of course, I have previously done so that we don't have to wait, and I can simply open the results. So as we have conducted a preliminary quantification, we get a count table here for the free samples of the parental line that tells us the number of supporting reads of each known as well as novel isoform that has been identified. We also get a report, again summarizing our analysis, as well as providing a little chart showing us the distribution of the length of our isoforms. So this step would now be repeated for the other 7 offspring lines in order to obtain, we can see it here, to obtain a GTF file with the transcriptome of each line. Now, since I've already done this, I can go ahead to the next step, which is, of course, combining all of these individual line-specific transcriptomes into one common sweet potato transcriptome. So, under the transcriptomics module, long-read analysis, and combining transcriptomes with TAMA merge, we can load all of our different line-specific transcriptomes as input data, as well as including the reference annotation in order to create a sort of extended reference annotation. We can also choose to give priority to the reference so that in case of any disputes between, for instance, the transcription start and termination sites, we can say that whichever annotation the reference contains has a little bit of priority over the others. We can configure our analysis in more detail, of course. Now here, by default, we use a splice junction threshold of 0, meaning that any transcript models that have any variation in their splice junctions will not be merged into a common transcript model. We keep this default because we sort of want to say that FLAIR is already taking care of defining these splice junctions for us. However, we are applying thresholds of 50 nucleotides for both the 5' and the 3' ends of our transcripts. So if any highly similar transcript models in, for instance, different lines are defined with slightly different transcription start or termination sites, they will still be merged into one common transcript model. Again, we define our outputs. We want to obtain the merged transcriptome as a GTF is the most important option here. And we're ready to launch our analysis, or in my case, jump ahead to our results. So we can see we have obtained a GTF file with the common transcriptome annotation for our sweet potato, and as always, a report summarizing our analysis. Now, of course, we don't know much yet about the structure of this transcriptome annotation. And this is where Scanty3 will help us out. So under transcriptomics and long-read analysis, we can find the option to curate long-read transcriptomes with Scanty3. Now here, as the transcriptome annotation file, we will be including the GTF file that we have just produced with TAMA merge. And we want to compare it to the same reference genome that we used for the reconstruction, as well as the reference genome annotation. Now, generally with Scanty3, we strongly recommend that any orthogonal data, such as short reads, such as poly-A peaks and transcription start site peaks, are provided in order to give the most information possible to your transcripts. However, working on a non-model organism, these data types are not always available. So today we will sadly not be able to include them. So here, no short read information, no transcription start site or poly-A peak information. We will be providing the preliminary quantification that we have obtained from FLAIR, that I have taken the time already to merge all of the different quantifications for each line into one common file, so that it gives us a sense of the expression level of our transcripts. Now, we also have different options to configure Scanty here. So as I've already mentioned, normally we would recommend using the machine learning filter, which can be very effective, especially when you have access to orthogonal data. But as we do not have such data for our analysis today, we will be using a set of custom rules. And we have customized our rule sets here in this JSON file that we will go over in 1 second. Further, we can also include the scanty-free rescue step, which is basically another pass over the transcripts that have been filtered out in order to try and find closely matching reference transcripts. So this is a step that is really trying to make the most out of your data so that you don't lose the reads assigned to the filtered out transcripts, but rather we can try to retain them by assigning them instead to a closely matching reference. So after configuring also our outputs, we're ready to launch our analysis. And of course, in my case, skip ahead to the results. Actually, first, I would like to discuss in a little bit more detail the filter that we have just introduced. As I have mentioned, we have a customized set of rules that we have used because we don't have access to orthogonal data. Specifically, the rules we have introduced are that we require a transcript to have a percentage of A's downstream of the transcription termination site of under 60%. Now, this is a fairly standard rule in scanty-free filtering that is essentially aiming to filter out any artifacts produced by intra-priming, by primers binding to A-rich regions within a transcript. Now, we also require transcript models to have an expression level or a number of supporting reads of at least 10, which simply helps us to get rid of some transcripts that are so lowly expressed that we might not be very confident in them being real. And further, we have introduced some rules to be a bit more strict about monoexons. So for monoexons, we either require them to lie within plus or minus 50 base pairs of the reference transcription start or termination sites. So basically, the monoexon has to closely match a reference monoexon, or it has, if it does not match a reference, it has to be predicted to be protein coding with a coding sequence length of at least 300 nucleotides. Now, once we have conducted our scanty-free analysis, we have to first understand the scanty-free structural categories, which, if you're not already familiar with them, I will take a short moment to explain them. So essentially, the goal of the scanty-free structural categories is to categorize and characterize the transcripts in your transcriptome when comparing them to the reference. So we can see here in this graphic, we have a reference transcripts, and then we have the 4 main categories of transcripts that we can identify. So a full splice match matches all splice junctions of a reference transcript perfectly. So there is no variation in the splice junctions. There can be some variation in the start and termination sites, but the splice junctions are the most important indicator of transcript identity, and those are equal to a reference. Now, an incomplete splice match matches the reference splice junctions only partially. So at either end or both ends, one or more splice junctions can be missing, but the existing splice junctions do match a reference transcript. Now, this can be genuine biology if we observe an alternative transcription start or termination site, or it can also be an artifact if it is, for instance, produced by RNA degradation. So we have to be careful with such transcripts. Now, the actual novelty in our transcriptome will mostly consist of the next 2 categories, which are novel in catalog and novel not in catalog. The green category, novel in catalog, is a novel combination of known splice sites. So if we look at this example, we can see our reference transcript has a small exon here, which is missing in our novel in catalog. So we have a splice donor and acceptor sites, which are exactly equal to what we observe in the reference, but the actual splice junction is novel. So we have a novel combination of known splice sites. Now, novel not in catalog isoforms, however, have at least one splice site that is completely novel, that is not contained in the reference. which in this example is located here. This exon is a bit longer as this splice acceptor site is a bit moved, and so this is classified as a novel not in catalog. There are many other categories, for instance, genic introns or genic genomic, but these four are really the main interest usually. So to move ahead to the results that we observe in OmicsBox, we can see for each transcriptome that we characterize and filter, we get a table with all sorts of information and details about each of our transcripts, and we get a variety of different plots, which are all customizable. But for our specific analysis now, since we're actually wanting to compare different lines, I've created a specific plot showcasing this. So again, we see the structural categories that we've just discussed, and along our x-axis, we have different transcriptomes. So on the right here, we can see we have our parental line followed by the 7 offspring lines. So for each of the transcriptomes that FLAIR has produced for us, I have conducted a scanty-free quality control analysis, and we can see the variety of different transcripts that we have in each of the lines. Now, when we use TAMA merge to combine them, we can see we get a quite large number of transcript models, which is the sort of the motivation for our filter, that we don't believe that all of these novel transcripts that have been discovered are genuine biology, and so we want to use scanty-free to curate them. And when we do so, we see that a very notable amount of the transcript models are actually being filtered out, which is, of course, not to say that it is in any way guaranteed that there are no artifacts left in our curated transcriptome. Neither are we guaranteeing that all of the things we filtered out are non-genuine, but at least it has applied our rules, which we could also, of course, go back and further adjust them. It has applied our rules, and it has given us a lot more confidence in the transcriptome that we have curated, in order to have a bit more trust in our results in any downstream analysis going forward. So now, moving back into OmicsBox for the final few steps of our analysis. Now, of course, that we have lost a lot of potential novelty in our transcriptome, we could simply move on with the same quantification we already have, and simply erase all of the filtered out transcripts. But that would sort of cost us a lot of information from our data. And of course, always we want to make the most out of the data we have. So by requantifying based on the curated transcriptome, we can try to save some of the reads that would otherwise be lost by assigning them or allowing FLAIR to assign them to a closely matching transcript instead. So again, under transcriptomics and long-read analysis, transcript identification, we will be using again FLAIR to now requantify our data based on our curated transcriptome. So this time we will not be running it once for each line. We will simply be using all of our data from all of our lines, providing the same reference genome. And now, important, we want to turn off the step of transcriptome reconstruction. So we don't want to discover any more novelty. We have already achieved this step. Now we simply want to quantify. In order to quantify, of course, now we need to tell FLAIR what its basis for quantification should be. Now, before we supplied here our reference transcriptome, which of course now we have to exchange for the curated transcriptome that we have produced. And like that, in this case, we don't need alignments, as the quantification happens based solely on the FASTQ files. We can configure our analysis again. For instance, if we wanted to be a bit more stringent, we could turn on some of these options. But for our purposes, the default settings are fine. We can configure where we want to receive our results, and we're ready to launch our analysis. So skipping ahead, we can see that we have obtained count tables. I have actually also aggregated the transcript level count table into another gene level count table. And now our results look like this. We have all 19 of our samples available in our quantification table with our curated transcriptome. So now that we have obtained a final quantification table, basically from here on out, it's a very standard differential expression analysis. So just to show you as an example, now thankfully all of our lines have at least 2 samples, so we're able to use the pairwise differential expression analysis with EdgeR. And we can configure our filters, upload our experimental design, which you can see we have our 19 different samples across 8 different lines. And we have a certain trait that in some of our lines is present and in others it is absent. And we might want to test these groups of samples against each other. So we can choose a simple design based on our trait column. As the contrast condition, we can choose that the trait is present, and the reference is the trait is not present. We conduct a robust exact test, and we're ready to once again launch our analysis, which will provide us with results such as this. A table full of all of our transcripts of our curated transcriptome with the results of our tests, and with tags indicating whether they're statistically significantly down or upregulated or neither. Now we also get a summary report which shows us, for instance, the number of down and upregulated transcripts. Now, if, for instance, we have identified, or we feel that we have identified too many, we can also use this action to set the up-down tags differently to, for instance, introduce a more stringent filter based on the false discovery rate or a more stringent fold change threshold. But for now, we are fine with our results. Now, instead of testing between our traits, we could also test between different lines, for instance, comparing the parental line to the offspring lines. We could also, this really opens a lot of doors into different downstream analyses. We could go into functional analyses. We could conduct a Fisher's exact test or a gene set enrichment analysis. We could look at pathways, but we have already covered these topics in previous webinars. So for today, I think it is time to move on to our conclusions, which are, well, we have conducted a long-read RNA sequencing pipeline on a non-model organism, all the way from alignments to differential expression. We have discovered novel isoforms with FLAIR, and we have handled multiple biological replicates by combining them with TAMA merge. We also conducted transcriptome quality control and curation with SCMT3 in order to improve our data quality. And we have then requantified our data based on this curated transcriptome and used this quantification to conduct differential expression analysis. Now, you've also seen that this entire process is made quite easy with OmicsBox. With just a few clicks, we go from raw data to results, and we can play around with parameters and inspect our results in a lot of detail. So these are the reference used for all of these different steps in our analysis. And with that, I would like to thank you for attending and for your attention. Now, we have recently published some blog posts about long-read RNA sequencing analysis, such as this one, where we dive a bit deeper into a workflow with FLAIR and SCMT3, or another blog post about a poster presentation of my research work, which I have mentioned before, that we also have a manuscript in preparation all about how to deal with biological replicates in long-read RNA sequencing data. So keep an eye out for that as well. And well, again, thank you, and I will hand the word back over to Stefan.
Thank you for watching. Learn more about OmicsBox with our other videos.